Hello everyone, this is Jujebalpiao from the Institute of Chinese Material Medical. It's very nice to give a talk on this great ginseng conference, especially in this special COVID-19 epidemic time. And I thank very much to my mentor and friend, Professor Yang Taijin, for his invitation. And my issue is about the ginseng cultivation and R&D using the methods of genomics. In China, the traditional ginseng cultivation model is in the forest-covered field. It's about for more than 500 years. As everyone knows, using this measure will give a great damage to the environment. Like in our photos, this is a bird eye view for the ginseng cultivation, and we can find that there is a very bare region after the ginseng harvest. So in China, from the 2019, even 1 acre forest field is not permitted for the ginseng cultivation. So for the whole industry, we should develop some new method for our ginseng product. Ginseng cultivation in farmland is a practicable alternative choice for our ginseng cultivation industry, as this method have been used in Korea and in Japan for hundreds of years, and currently the GIS method and other modern technology give us a lot of help for the ecological factor evaluation during the field choice. Except the environment issues, our ginseng cultivation industry is also needed for the upgradation in some other aspects, like the pesticides and the fungicides, because some of the farmer for ginseng is not very familiar with the pesticides used, so they will applicate a lot for the ginseng cultivation. And in the food, most of the countries have used the standard of pollution-free. So we think introducing the pollution-free standard of process will help the ginseng upgradation. And Our group is good at the genomic research, so we from the genomic view and establish a full system for the ginseng cultivation in farmland as the G-scattered field selection and using the pollution-free standard. And we use, based on the genome, we use the marker-assisted breeding, and during the operation, we supervised the farm to follow the pollution-free SOP, and for the soil improvement, we using the metagenome to investigate the soil and establish our manual combination. We finished our genome map for the ginseng. At that time, ginseng is one of the most complex plant genome. More than 60% of its sequence is from the repeat content. And we also use the daisy mass spectrometry imaging for the ginseng node size distribution analysis, and using based on the imaging results, we separate the ginseng root to different sub-organ parts and send for the transcriptome analysis. And according to the genome and the transcriptome data, we identified the upstream pathway and their paralogs involved in the backbone synthesis in ginseng. I think the number is about 31 for both the genes and their paralogs. And we also identified more than 200 UGT for the metabolite diversity. As the UGT will transfer the glycobase for the ginseng node size backbone. And then we use the key genes as a marker for the ginseng cultivation. One of our example is that we used the HMGR and the SE genes for the selection of the suitable manual graduations. We also analyzed the origins and their regulation. This paper has been selected as the elect paper of Chinese Academy of Sciences. BHLH is a very important transcriptional factor in plants. So we also identified their family in its family, the family in ginseng, and we found some of the genes have the high correlation with the expression of the key enzymes involved in the ginseng-noside biosynthesis pathway. The OPT family is a group for the electrochemical potential-driven transporter that catalyze their solutes in an energy-dependent sympathetic manner, may play important roles in plant growth and abiotic and biotic stress. We identified 37 from OPTs from ginsengs, and these genes were clustered into two distinct clades in which clade-specific motif compositions were considerably conservative. The distribution of OPT paralogs was indicative of segmental duplication and subsequent structural variation. And plant hormones act as chemical messengers in the regulation of plant development and metabolisms. We also identified the PIN and PIN-like auxin transporters. In this study, identification and transcription profiling of PIN and PILS genes families, as well as their potential relationship with ginseng-noside biosynthesis, were studied. A total of 17 PINs and 11 PILS genes were found. Sequence alignment, positive motif organization, and subcellular distribution were analyzed. Furthermore, correlation analysis indicated the biosynthesis pathway for ginseng-noside showed the Pearson correlation coefficients of 8 PIN over PILS genes. And except the basic analysis, we also set a system for the application of our genome in the marker development and the marker-assessed selection breeding of medicinal plants. And first, we will identify the distinct phenotype of the plants and found their correlated markers. Then we use this marker further for the select of the population. Now, based on those population, we further breeding our breeding our species. For ginseng, we have established different populations in our pasture wormhouse. Like the stem, some have the purple stem and some have the green stem. And for their fruit, some have the red, some have the yellow. Then we sequencing them and to find some markers. Using these markers, we further to detect the isolates and gather them together to form a special group. And based on this group, we select our breedings. And for the cultivation, we also set our SOP, and like the soil improvement, we use the green manure and the farmyard manure. And we also identified the pests and fungi that are easily for the infection of ginseng, and we select suitable pesticides. And we also established a framework for the ginseng quality tracing from the seed to production, as used the DNA barcoding for the species control, and using the chemical finger mark for the quality identification. And in our server, we will record all the information in the whole production chain, and to ensure the quality of the ginseng production. Now we have got a lot of achievement for our ginseng work, as we released our ginseng genome, and at the same time, Professor Yang released theirs, and I and the Professor Yang co-edited the ginseng genome book. We identified a lot of key enzymes for the ginseng nosed biosynthesis, and now we have got two certificated breedings for our farmland cultivated ginseng. And our base is more than 3,000 acres, and we have our special manuals, and we also put forward our standard for the ginseng pollution-free ginseng cultivation, and this standard have been certificated by the China Academy of Traditional Chinese Medicine, and we also published our book for the ginseng cultivation. These works have been awarded by the Chinese Academy of Traditional Chinese Medicine, and also have been certificated by the Jilin province. Except the cultivation, we also used our omics tools for the ginseng nosite development. One is for the prebiotic fibers for the ginseng nosites. We all know that gut microbiota mainly function in the biotransformation of some primary ginseng nosites into the biotic form. Herein, we investigate the effect of three prebiotic fibers. One is the food oligosaccharides and the galactic oligosaccharides and the 5-2 by targeting the gut microbiota and metabolites of ginseng nosites RB1 in vivo. Sprague-Dawley-Rice were administered with ginseng nosites RB1 after a 2-week prebiotic intervention showed that the peak plasma concentration and area and concentration time curve of ginseng nosite RB1 and the intermediate metabolites such as the ginseng nosite RD, F2, and CK. All these groups were increased at a various degree compared with those in control group. Meanwhile, the gut microbiota dramatically responded to the prebiotic treatment at both taxonomical and the functional levels. The abundance of the prevotella, which possess potential function to hydrolyze ginseng nosite RB1 into CK, was significantly evaluated in the three prebiotic groups. The gut metagenomics analysis also revealed that the functional genes enrichment for terpenoid metabolism. These findings imply that prebiotic may selectively promote the proliferation of certain bacterial strains, thereby subsequently improving the biotransformation of some certain ginseng nosites. So this will be very benefits for the development of ginseng foods. And another work is about the AFLD treatment with ginseng nosites. In addition to the pharmacokinetics, we investigated the possible role of gut microbiota in the pharmacological efficacy of ginseng nosites. The major ginseng nosites exert benefit effect on the non-alcoholic fatty liver disease due to the multi-component and multi-target features of ginseng oxide. Their detailed mechanisms remain unknown. So in this study, we used the C57BL mice were fed at a high fat diet supplement or not supplement with ginseng oxide exert for about 3 months. Ginseng oxide extract significantly elevate the high fat diet, induced the non-alcoholic fatty liver disease. In a dose-dependent manner. Furthermore, ginseng oxide extracts treatment the modulate and HFD-induced imbalance in the microbiota and alleviate the dysbiosis mediated gut leakage. A network analysis revealed that the anti-inflammatory effects and the regulation of the metabolic balance might be responsible for the effect of ginseng oxide extract on this disease. These findings implied that the benefits of ginseng oxide extracts that for the NFLD disease. And this except ginseng, we also use our omega tools for other ginseng genus species like the Panax quinquefolius and the Panax noto ginseng. And we also introduce some new chemical analysis tool for ginseng development as the RIMS, which use a knife-like tool. In this top, it can be quickly heated to a very high temperature, and the chemical can be detected by the mice system and give authentication in a very fast speed. My introduction of our work. Thank you very much for listening.